Welcome back to the deep dive. If you've ever looked at a textbook diagram of the tree of life and wondered, how did we get from simple chemicals to, well, us, then this deep dive is definitely for you. We are jumping right into the core source material for the history of life itself.
That's exactly right. Our mission today is really to summarize that incredible evolutionary journey. It's a multi-billion-year saga, all detailed in this chapter's core concepts. We're basically tracking the sequence,
Right.
starting with how the planet formed, then moving through those absolutely critical metabolic shifts that change the atmosphere forever, and then finally looking at how those energy upgrades just fundamentally rearranged our whole system for classifying life.
And we have to start, well, literally at the beginning, 4.6 billion years ago when Earth first coalesced. We'll chart those first two non-biological atmospheres and then get into the great oxygen flood that basically created the world we breathe in today.
Yeah, and we'll use the evidence, the stromatolites, those banded iron formations, and the molecular fossils to really anchor the timeline. Then we'll dive into the metabolic breakthrough. The staggering energy difference between that ancient anaerobic life and modern aerobic life, and how that sheer power fueled the rise of complex cells and the revolutionary classification system scientists now use.
Let's unpack that first environment then. When Earth formed, there were two distinct atmospheres, right?
Yeah.
And the fascinating thing is life had nothing to do with either of them coming or going.
Correct. The first one, the primary atmosphere, didn't stick around long, mostly hydrogen and helium. But Earth's gravity was, well, just too weak. Solar winds were intense. The planet was incredibly hot.
So it just got stripped away.
Pretty much, yeah. Gone within maybe 500 million years after formation.
Wow. Okay, so then came the secondary atmosphere. Around, 4.2 to 3.8 billion years ago?
Yeah, that's the one. This one came from inside the planet volcanic outgassing. Basically a hot mix of water vapor and carbon dioxide. But the key thing, almost no free oxygen. Zero for all practical purposes.
And that lack of oxygen is everything, isn't it? Because the first life that showed up, maybe 3.8 billion years ago, had to survive without it.
Exactly. These were single-celled methanogens, methane generators. They're strictly anaerobic. Their whole metabolism runs without oxygen. And they're waste, methane.
Yeah, and it's amazing how we know they were there. Not just fossils of cells, but molecular fossils.
Yeah, it's incredible. Researchers found methane signatures, basically chemical fingerprints that are definitively biological, trapped inside super old rocks. Specifically, silica dikes in Western Australia dated over 3.5 billion years old.
So actual chemical proof of ancient microbes.
Precisely. And this methane, their waste product, actually did something vital, though completely by accident. It acted as a really powerful greenhouse gas. In that oxygen-free atmosphere, it formed this insulating layer that kept Earth warm enough. It probably prevented a global freeze-over around 3.5 billion years ago. Life's waste literally saved the planet from becoming an ice ball.
That's wild. And we see physical proof of that in anoxic world too, right? Like in old riverbeds.
Definitely. You find these 3-billion-year-old river deposits, and the sand isn't normal sand. It's made of iron, lead, and zinc sulfides. Sulfides like that just, well, they can't form, or at least they can't last. If there's significant oxygen around, they'd oxidize.
So the rocks themselves tell the story of no oxygen.
They do. So we went from this methane-warmed, oxygen-poor world right into what must have felt like a total metabolic catastrophe for the life forms back then. Because here's where the revolution happened. Some organisms evolved the ability to use light energy to make food. Photosynthesis.
And the byproduct.
Oxygen. O2, a poison to most existing life.
Right. And at first it didn't really build up in the atmosphere, did it? It just reacted with rocks and stuff in the water.
Exactly. It took a very, very long time. Oxygen levels stayed really low, like maybe 1% of what we have now, until roughly 2 billion years ago. Geochemists think the big rise started maybe 2.3 to 2.0 billion years ago, climbing towards today's 21%.
And the organisms responsible for this were the cyanobacteria.
They were the key players, the first cells capable of oxygenic photosynthesis. That means using easily available water as the source for electrons and hydrogen. And that process releases O2 directly. It forced the planet to become aerobic.
And we have fossils of them, and these, stromatolite.
Oh, yeah, remarkable evidence. Stromatolites are these amazing layered kind of rocky mounds. They're built by mats of microorganisms trapping and cementing sediments, the oldest cyanobacteria fossils. And these stromatolite structures, they go back 3.5 billion years. There's this famous reef in the Strelley Pool Chert in Australia 3.4 billion years old. Just incredible.
It's funny, you only find living stromatolites today in really harsh places, right? Like super salty lakes.
That's true, because in normal environments, things like snails would just graze them down. But back then, those kinds of herbivores hadn't evolved yet, so cyanobacteria could build these massive structures almost everywhere shallow water existed.
Wow. And if you look inside one of those ancient mats,
You see a whole community. The oxygen-producing cyanobacteria are right at the top, getting the most sun. But underneath, you find other photosynthesizers, like green sulfur bacteria, which don't make oxygen. They use different wavelengths of light that filter down. It's like a little layered ecosystem.
and another piece of evidence for all this oxygen flooding out, the banded iron formations, or BIFs.
Ah, BIFs, yes. These are really distinctive sedimentary rocks. Some date back almost 3.8 billion years. They have these characteristic alternating bands, usually reddish iron oxides like hematite and magnetite, layered with shale or chert.
So how did they form?
Well, think about all that new oxygen being pumped out by cyanobacteria into the oceans. The oceans back then were full of dissolved iron, ferrous ions. The oxygen immediately reacted with that dissolved iron.
Making rust, essentially.
Basically, yeah, insoluble iron oxides. These precipitated out, rained down onto the seafloor, and over vast amounts of time built up those distinct layers. They're a direct record of oxygen hitting iron-rich seawater.
Okay, that's compelling. There's another type of evidence too, right? Something about carbon isotopes?
Yes, the carbon-13, carbon-12 ratio. It's a bit technical, but basically living organisms prefer to use the lighter isotope of carbon, C12, because their enzymes work a bit better with it.
Okay.
So carbon that's been processed by life has a distinct isotopic signature. It's relatively enriched in C12 compared to C13, and guess what?
They found it.
They found that signature in some of the absolute oldest sedimentary rocks known, from the Isua Greenstone Belt in Greenland. Rocks dating back 3.8 billion years. It's independent proof that life was already active way back then, even before the oxygen really started building up.
Wow. Okay, this all brings us to the huge difference in how life runs, the metabolism. The rise of oxygen didn't just change the planet's chemistry, it changed life's engine. Let's unpack that energy upgrade.
Right. The original way, the ancient way, the almost universal pathway, is anaerobic glycolysis. It breaks down glucose into pyruvic acid without any oxygen. It works, it's essential, but the energy yield is tiny. A net gain of just 2 ATP molecules per glucose.
ATP being the cell's energy currency. So 2 units, that's not much.
Not much at all. It's functional, but maybe like the efficiency of a hand drill compared to a power tool.
And the enzymes involved in glycolysis are basically the same everywhere, right?
Yeah.
From bacteria to us.
Highly conserved, yeah. It tells us this pathway is incredibly ancient, a shared inheritance. It wasn't just random, it was channeled by what chemicals were available and what worked early on.
So what was the first step up from that? Using light.
Exactly. Early photosynthesis, often what's called cyclic photosynthesis, used light-absorbing pigments to generate a bit more ATP. But the real quantum leap needed 2 different chlorophyll systems working together. System I and II, that allowed for non-cyclic photosynthesis.
And that's the one that splits water.
That's the one. 2H2O gets split into electrons, protons, and crucially, O2. That's the oxygen source again. But having that oxygen around opened the door for a whole new level of energy production. Aerobic metabolism.
Using oxygen.
Using oxygen. This new system uses the Krebs cycle, or citric acid cycle, and oxygen acts as the final acceptor for electrons in this really complex, membrane-bound electron transport system.
Okay, and the energy payoff, that's the key part.
It's absolutely staggering. Complete aerobic oxidation using oxygen yields maximally around 38 ATP molecules per glucose.
38. Compared to just 2 from the old way.
Exactly. An 18-fold increase in energy efficiency, that massive amount of available energy. Well, that's what paid for building bigger, more complex cells.
Which brings us neatly to the 2 big divisions of life, prokaryotes and eukaryotes.
Right. The prokaryotes came first. They're small, typically 0.5 to 10 micrometers. They lack a proper nucleus membrane, don't have complex internal organelles, and they divide by simple binary fission. They showed up maybe 3.8 billion years ago.
Then much later, around 1.8 billion years ago, the eukaryotes arrived.
Yep. And they are different, much larger, 10 to 100 micrometers. They have that defining nuclear membrane, a complex internal skeleton, those powerhouse organelles like mitochondria, and they divide by mitosis.
And that energy difference we just talked about, the 2 ATP versus 38 ATP, that's what likely funded all that extra eukaryotic complexity.
It's almost certainly a huge part of the story. You need that energy surplus to build and maintain all those intricate structures.
Okay, so as we started understanding these fundamental cell differences, our whole classification system had to change, didn't it? It wasn't just plants and animals anymore.
Not at all. For ages, yeah, it was Plantae and Animalia. But by the 1940s, scientists like Stanier and Van Niel really emphasized the huge gap between prokaryotes and eukaryotes.
So those became the two super kingdoms.
Initially, yes, though they noted both groups were probably polyphyletic, meaning they didn't each arise from a single common ancestor within the group. Then came Whittaker in 1959 with his 5 kingdoms, 1 prokaryote kingdom, Onera, and 4 eukaryote kingdoms.
But the really big shakeup was later, with genetic analysis.
Oh, yeah. The 1980s, Carl Woese and his colleagues using ribosomal RNA analysis. That just blew the old system apart.
Leading to the 3 domains.
Exactly. The 3 domains of life, Eubacteria, your standard bacteria, Archaea and Eukarya, us and all other complex life, and the bombshell was the Archaea. They look like bacteria, but genetically, they're distinct from Eubacteria and actually share some core molecular features with us eukaryotes.
So Archaea are kind of our distant cousins, not just weird bacteria.
That's a good way to put it. It fundamentally redrew the base of the tree of life.
And things get even more interesting within our own domain, Eukarya, right? We're moving beyond those old 4 eukaryotic kingdoms like Protista.
Absolutely. Protista is, frankly, a mess. It's just a grab bag of single-celled eukaryotes that aren't plants, animals, or fungi. It's wildly polyphyletic and doesn't reflect real evolutionary relationships at all.
So what's the current thinking? Supergroups.
Exactly. Modern classification uses 5 or 6 eukaryotic supergroups. Things like Excavata, which includes some nasty parasites like Giardia that causes hiker's diarrhea, and Trypanosoma, which causes sleeping sickness, or Chromalveolata, a huge group containing everything from giant kelp forests down to Plasmodium, the parasite that causes malaria.
Okay, diverse groups. But you mentioned one that's particularly surprising.
Yes, the Uniconta supergroup. This is the one that always makes people do a double take. This lineage includes animals, fungi, and some single-celled relatives called choanoflagellates, all grouped together.
Wait, animals and fungi together.
Together, specifically within a lineage called the Opisthocons. So evolutionarily speaking, you are much more closely related to a mushroom than you are to a rose bush or a piece of kelp.
That is definitely counterintuitive.
Isn't it? It really highlights how molecular data has reshaped our understanding.
Okay, so this constantly evolving classification, this picture of increasing cellular complexity, it brings up a big question, doesn't it? Has complexity actually increased over evolutionary time?
The complexity question, yes. It seems obvious, right? You look at a bacterium versus a blue whale and say, of course complexity increased. But evolutionary biologists are often hesitant. They tend to shy away from using terms like progress or even definitively stating complexity always increases.
Why the reluctance? It seems pretty clear.
Well, part of it is defining complexity. It's actually really slippery. What are we measuring? Genome size? Not always reliable. Some amoebas have way bigger genomes than humans. Gene number? Again, not a perfect correlation. Cellular compartmentalization. Number of protein interactions.
Like the human protein interactions you mentioned.
Yeah, humans have an estimated 650,000 interactions between proteins, which is way more than fruit flies or nematodes. But what that means in terms of an objective scale of complexity, it's still pretty enigmatic.
But you said there is one metric that seems...
One measure that seems less ambiguous and generally points upwards, at least in animals. The increase in the number of distinct cell types.
Okay, tell me more.
If you track the evolution of animals, the number of specialized cell types consistently goes up. Sponges might have, say, 6 to 12 types. Flatworms, maybe 20 to 30. Mollusks and arthropods, around 50 to 55. Humans, somewhere between 200 and 400, depending on how you count.
So that specific measure does support increasing complexity, at least for part of life's history.
It does. It's probably the most widely accepted metric supporting that conclusion, based on work by Valentine and others back in the 90s.
And it also helps clarify what not to use as a measure, right? Like just being bigger.
Exactly. Organism size isn't a good proxy. Cope's rule suggested lineages tend to get bigger, but there are tons of exceptions, especially parasites, which often get smaller and simpler structurally. And life history complexity, like a tapeworm's incredibly complex multi-host life cycle, doesn't mean the organism itself is more complex at a cellular or organizational level.
Right. Okay. So summing up then, we've traced the shift from an anoxic world shaped by volcanic gases to an aerobic one, terraformed really by life itself through photosynthesis.
Driven by cyanobacteria, yeah. And that led to the massive metabolic upgrade from a measly 2 ATP to a powerful 38 ATP per glucose molecule.
which fueled the rise of eukaryotes in all subsequent complexity. And our understanding of life's relationships has been revolutionized by molecular data, rearranging everything into domains and supergroups.
Showing us surprising connections, like us and fungi being evolutionary siblings in the Uniconta.
It really is a journey fueled by energy and mapped by chemistry and genetics. But as we wrap up this deep dive, maybe a final thought for our listeners, building on that complexity idea.
Okay, well, consider this. If the increase in specialized cell types marks a major evolutionary transition, as many biologists think, what fundamental transition might be happening now, maybe related to information processing or global ecological integration, or something we haven't even conceived of yet? What shift, looking back billions of years from now, would future biologists see as the next major, irreversible step in life's complex unfolding?
That is definitely something to ponder. What's the next big jump? Fascinating. Thank you so much for walking us through the absolute foundations here.
My pleasure. It's always amazing to revisit the sheer scale of this story.
Until next time, keep exploring those fundamental questions.